Okay, so gene expression. Gene expression is what we're talking about this last part of chapter 3, and that's how we go from a gene, our DNA, genetic material, to a functional protein. So for this, we're going to be looking at the difference between what DNA transcription is and translation. We'll talk about chromatin, and we had talked about chromatin in the last section, and I told you that it was that more loose form that you find during interphase whenever cells would be making proteins, because it's still coiled tightly, but it's a little bit less coiled. But there are two different types of chromatin, euchromatin and heterochromatin, so we're going to look at the difference between them. And we'll generally describe the structure of chromatin and how proteins called transcription factors will adjust the structure. We'll also talk about how RNA is made from DNA and the different types of RNA. We have pre-messenger RNA, messenger RNA, ribosomal RNA, and transfer RNA. And then we'll talk about protein synthesis. And lastly, we'll talk about DNA replication, which is pretty easy considering we talked about it right there. It's just where DNA makes a copy of itself. But first, gene expression, which includes the two processes, transcription and translation. Genes are lengths of DNA that code for how to make a messenger RNA molecule. I mean, generally, genes are lengths of DNA which will code ultimately for a protein, but initially they are lengths of DNA that code for messenger RNA synthesis. Interestingly, we have 25,000 actual coding genes. There is actually a lot of DNA that is not coding. We have just nonsense is what they call it. It doesn't code for anything. But we do have, mixed among that, we have 25,000 actual coding genes. Here, this is very interesting. Human DNA, being that humans are all the same species, we are 99.9% genetically identical. That's why all humans have the same things on their face: a nose, 2 eyes, a mouth, hair, 2 ears. The body plan is pretty similar, although sometimes people have, a heart on the opposite side or something like that. But only about 0.1% of DNA is what makes us look different. So some people are more easily able to acquire muscle. Some people have different colored eyes, different kind of hair, different skin colors, all of that. But that's why all humans can reproduce, because we're genetically very, very similar. So messenger RNA is the first type of it's the first kind of RNA that we're going to produce, and it does carry the information on how to make a protein. So to express a gene, to go from DNA to a protein, there are 2 stages. We have transcription, which is where we take our DNA sequence and turn it into a messenger RNA sequence. And that's going to occur specifically in the nucleus of the cell. The other process is translation, and translation is where we go from messenger RNA to our protein. And this occurs on a ribosome, either in the rough ER or the ribosome in the cytoplasm. And this is going to happen outside of the nucleus, somewhere in the cytoplasm, again on an organelle or on a ribosome within the cytoplasm. Each nucleus contains one or more dark areas called nucleoli. The nucleolus of a cell makes ribosomal RNA, which is going to be associated with the ribosome and is the part of the ribosome where the protein synthesis takes place. Protein synthesis, also known as translation. So a couple of terms here. genome and proteome. A genome specifically refers to all the genes that an individual has, or all of the genes that a particular species has. Again, like human beings, the way that we look, we have 2 eyes, a nose, and a mouth, regardless. That's all in our genome in terms of how we're going to be made. A proteome refers to all the proteins that you can make from all of those genes in the genome. Even though we have 25,000 coding genes, we can make over 100,000 different proteins because one gene, and we're going to see how, can code up to 3 proteins, not just one. So again, we're going to see how that happens. But first of all, chromatin, as I mentioned earlier in the class today, is DNA, which is highly ordered and organized, but it is the more loose form out of chromosomes and chromatin. So what we're looking at here, this is kind of unraveled, it's made up of little proteins called histones, which are a lot like a spool around which the DNA will wrap twice. And DNA is like thread that goes around the spool. Together, the histone protein, which is in yellow here, and the DNA wrapped around it in purple is known as the nucleosome. So the histone and the DNA together is called a nucleosome. Histones are positively charged and form spools around which the negatively charged DNA strands wrap. That's what attracts the DNA to the histone protein, are the opposite charges that they have. And we have about 5 nucleosomes in each row on the DNA molecule. So this DNA is very, very tightly wound, and this is the chromatin form. Whenever we go into mitosis or meiosis, the cell division phase, this DNA supercoils into chromosomes. Do you have any questions about that?
You said there's 5 spools.
I think of the nucleosomes. Yeah, of the nucleosomes. Any other questions? All right, so there's 2 forms. There are 2 forms of chromatin. There's euchromatin and heterochromatin. Euchromatin is the part of the chromosome that's actually active in transcription. So it is going to be sort of a little bit less coiled so that we can get to the gene inside. So this is going to be light in color. It's not as dense. Heterochromatin is highly condensed, in which the genes are hidden and you can't access them. So, for example, this is our, we can say this is a heterochromatin form here, where the genes are not easily accessed. But then through a process called acetylation, acetylation will open up that heterochromatin a little bit more into the euchromatin form so that the genes are accessible. Something called a transcription factor will bind to this, and ultimately that gene will be written down or transcribed into a messenger RNA molecule. When transcription is complete, deacetylation occurs, so we take away the acetyl group and it becomes heterochromatin again. Any questions about that? Heterochromatin versus euchromatin? Nothing?
I mean, I do, but it's really obscure.
What?
All these diagrams look like telephone.
Like a telephone?
Like the old, I can't think of what it's called. Like, never mind. It's irrelevant.
I don't see any telephone.
Like the way they used to wire up communication lines back when telephone lines first came about.
I don't even know.
Like the line wrapped around the transistor. said it's obscure.
I can't even visualize that. I'll have to Google that. I don't know. All right, so RNA synthesis. So one gene is usually several thousand nucleotide pairs long. DNA in a human cell contains over 3 billion base pairs. So it's really, really a lot, but only a fraction of that DNA actually has relevance. A lot of the DNA that we have, as I said before, doesn't code for a protein. It's nonsense. So what happens is when we transcribe a gene, which again, where does this happen in the cell? Transcription, where does it happen? Where do we, in the nucleus, we transcribe our genes. So an enzyme called RNA polymerase will bind to the start sequence on DNA and unzips the strands. So it's kind of like a Ziploc baggie, you know, where with transcription, you're not unzipping the whole bag. You're only unzipping a portion of it, just so that you can access what you need inside. That's essentially what you're doing with the DNA during transcription. We're only going to unzip a portion of the DNA molecule. We have promoter regions nearby, which regulate levels of transcription. And then we have what are called start and stop sequences, known as codons, where the transcription should start and end. RNA polymerase, that enzyme, will stop unzipping the DNA when it gets to the stop sequence. Transcription factors have to bind to areas on the DNA to initiate transcription and activate the gene. So over here we're showing the process of transcription. The purple strands is our double-stranded helix of DNA. And again, we get RNA polymerase unzipping the DNA, breaking the hydrogen bonds between those base pairs. And the blue strand that you see here is our messenger RNA molecule that's being produced. So it's single-stranded, and what we're doing is we're pairing the complementary bases with that gene sequence on DNA. So if DNA contains cytosine, our messenger RNA will contain guanine. If our DNA contains adenine, our messenger RNA is going to contain uracil, not thymine. Remember that RNA contains uracil, not thymine. If we have thymine, which they don't show it here on DNA, what's our messenger RNA sequence going to have? Adenine, right? Good. Okay, so with RNA, we actually have 4 different forms. The first form is pre-messenger RNA. This is a form of messenger RNA that is unedited, so it's going to contain some of the coding information we need, but also it's going to contain the nonsense we don't need. So we're going to talk a little bit about how that stuff is released from the messenger RNA molecule. Once we edit our pre-messenger RNA, now we have our messenger RNA molecule, which has just the sequence to code for our protein. Transfer RNA is going to bring amino acids, so it transfers amino acids to our growing protein. And ribosomal RNA, as we said earlier, is produced in the nucleolus and is the site on the ribosome where the protein is made. So first, as I mentioned, when transcription occurs in the nucleus, we first start with pre-messenger RNA. This is longer than messenger RNA and contains introns, which code for nothing, and exons, which are coding regions. So the introns are going to be released, and the exons are going to be what we call spliced together or glued together. Now, if you remember earlier, I said that we can, out of 100, I think there's 25,000 coding genes, right? But we can make 100,000 different proteins out of 25,000 coding genes. Why? Because there are alternate ways that these parts of the messenger RNA can be spliced together. So if we take out these introns, these dark blue areas, we could take this middle piece and splice it to the end, and this end piece we could splice over here. Or we could take this end piece and splice it over here, but we could mix it around, and so one gene could actually make different proteins from that one gene by the process known as alternative splicing. Alternative splicing. Human genome has 25,000 coding genes, but produces 100,000 different proteins because one gene can code for an average of 3 proteins because of alternative splicing. Okay, so protein synthesis occurs in the cytoplasm or in the rough ER on a ribosome. One amino acid is going to be linked to another amino acid one at a time, according to the sequence of codons in messenger RNA. In the cytoplasm, when the messenger RNA attaches to the ribosome, we call this a polysome, and that is where the protein synthesis will occur, or translation, as it's known. Okay, there's one correction I wanted to make here, and that is on DNA, we have 3-letter sequences that are known as a triplet, okay? So on a DNA molecule, we have a triplet. On RNA, 3-letter sequences are known as codons. Okay, so make sure you write this down because it's a little bit, in the notes it's a little bit long. DNA, 3-letter sequences are a triplet. In messenger RNA, 3-letter sequences are called a codon. On tRNA, transfer RNA, 3-letter sequences are anti-codons. So we have triplets on DNA, messenger RNA has codons, tRNA has anti-codons. Now on DNA, we have the 3-letter sequence, the triplet TAC. So our messenger RNA codon that is going to be complementary to that is AUG. And that codes for the start amino acid known as methionine. Methionine is pretty much always the amino acid that starts the protein. So you should probably write that down too. You don't really have to know the rest of the codons for the other amino acids, but methionine you should know. Yeah. It's the start, so it's the first amino acid on a protein, pretty much all the time. Okay, and then, let me just see here. Yeah, okay. So this is our DNA, this is our coding strand. This is showing us transcription. This is a nice diagram to show both transcription and translation. So this is our coding strand, our triplet TAC. Messenger RNA has the codon sequence AUG. And then the anticodon sequence not shown here is on transfer RNA. But ultimately, in translation, messenger RNA on the ribosome, that codon will code for methionine, which is the first, always the first amino acid. And then all the other amino acids are going to be bound together through peptide bonds, which are nonpolar covalents. This last thing just shows us the structure of transfer RNA, which looks a lot like a 3-leaf clover. On one end, it has its anticodon sequence. On the other end, it has an amino acid, and there's really no relationship between the anticodon. The anticodon is going to bind to the codon on messenger RNA, and that's how the transfer RNA knows where to bring the amino acid. It's essentially what it is. All right, so I have a video here that I liked. I thought it was a pretty good video, but I found this one, which I think is even better, just showing us what happens with protein synthesis. And after this, I'll let you go. But I wanted to finish this so it's not like strange, because it's a whole process. All right.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
What's the anticodon on the
This is added
transfer
to a
RNA?
growing chain of amino acids.
And that's the codon on the messenger RNA.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Primary, secondary, tertiary level protein folding. And that's it. If you have any questions, please feel free to email me, and I will get the study guide out before I leave today. And have a good holiday. Be safe. And yeah, I think on Tuesday, like I said, if there's any questions that you have, I can answer them before class. You can email me, I'll answer them over email. If you want to do like a virtual office hour thing, we can do that too. It's up to you.